Hi, everyone. Today we are going to be talking about something that we've all become very familiar with, which is SARS-CoV-2, or the coronavirus. COVID-19 is the disease that is caused by this virus. And we are going to take a look at how strains of the virus have evolved over time. This case study that we're going to use was published back in May of 2020, and so the data is a little outdated. It does not include the more recent strains that we have been seeing recently, from notably from South Africa and from Brazil, et cetera, that are now here in the United States. However, the tools that we're using, the BLAST tools, will include that. And so some of the questions from the case study might be answered differently than they would have been last May. Um, that's just a demonstration of how cool cooperative science is, because this data has been pouring in from around the world over the past year and has continued to do so. And it also shows how variable viruses are. Viruses, they mutate really quickly. We talked about mutations during our genetics unit, our molecular genetics unit, and we found that mutations can cause different versions of a gene or different alleles. In viruses, if they mutate in their genome, it can cause different variants of the virus. Some of them aren't very useful, some of them are useful. And so we're going to investigate a few different strains, and also geographical data for where the different strains are located and how different countries are interacting um, in terms of COVID-19. And so thinking through these problems will be really interesting after the year that we've had. And so go ahead and open up your case study. I am going to demonstrate kind of the first few steps. And so go ahead, open up your case study, open up NCBI, and if you have trouble with that, come on back. So I have navigated to the National Center for Biotechnology Information. NCBI is a wonderful resource, and I used it a ton when I was doing my bioinformatics class in college. This is where all of the DNA data that the world has lives. And so I've typed in severe acute respiratory syndrome coronavirus 2, which is SARS-CoV-2. That's the virus that we're working with. I'm going to take all databases and I'm going to click just genome and hit search. And I'm going to try to identify the ref-seq number. And so that's the reference sequence. And so if you look down here, you'll find it, NC underline 0455. Perfect. And so that's that first step. Okay, so now I have navigated to BLAST, the US National Library of Medicine. The BLAST tools are basic local alignment search tools. And so they are going to take sequences and compare sequences of DNA against each other. And you could either do DNA or you can do proteins with amino acid sequences. And so we are going to do a nucleotide blast. It's important to note you can also go from nucleotide to protein or from protein back to nucleotide. And so we're going to do a nucleotide blast. And we are going to type in the ref-seq number. And so I grabbed that from the last page from here. I have the ref-seq, and I am now going to blast that. Before I blast, I have to make one quick alteration. And so I'm going to go down here to algorithm parameters, and I'm going to change the max target sequence to 500. So that will just be a little bit more inclusive of different viruses. And so now I'm going to click blast, and it's going to work. This could take a little bit just because we're searching full genomes here. All right, so my blast has completed. And so if I scroll down, I can see all of these different matches for the sequence that I've identified. You can see different scores. And so there's max score, total score, query cover, e-value, percent identical, accession lens. And so these are different markers for how good the match is. You want to scroll down quite a bit, see how different they can get. You can see there's a lot of data. This is what I'm talking about when I'm saying that like everything from the past year has gone in here. And so take a look at this data, try to figure out what these things mean on your own, answer the questions, and then come back and press play on me. Okay, so hopefully you've answered the questions, written down your answers in a form where you can submit it. And now let's talk through them. So the first question asks about the geographical location of this data. And so if we look at our first few entries, we have the first one from Wuhan. Then we can see a bunch from the US, one from France, one from EGY. I'm not sure. Maybe Germany? Maybe England? We'll figure out what EGY is later. But so they came from a few different places. You can see down here, we get some from China. And so that is kind of where that data comes from. The values provided are a really interesting thing. And so the values that we're talking about, the first one is the max score. And so we see that here. The max score is the highest alignment score calculated. And that's basically from the sum of matched nucleotides or amino acids minus the penalties for mismatches and gaps, so areas where there wasn't a match. This is something that is kind of produced by NCBI and is a relative score. And so it doesn't mean anything about the sequence size or the length. It is a relative score for how well the two sequences match. And so the sequence that was found here and the sequence that we put in. The total score is the sum of the alignment scores of all of the different segments of the same subject sequence. And so they've split it up into different segments. Some parts match really well, some parts don't match so well. And so taking all of the segments and piecing them together, what is the total score then? And you can see that the max score and the total score here are the same because the max score is covering pretty much the entire sequence. And you can see that with the 100% query cover. What query cover is the percent of our search that was included in the aligned segments. The E value is the expect value. That's what the E stands for. It's the number of alignments that are expected by chance to just have happened accidentally, or that would happen like if you threw 10,000 nucleotides together, what are the chances that some of these would match? And so what are the chances that this happened by chance, that this was not a variant of the same virus, but that it just randomly happened to match? You can see that our scores here are 0. And so this means that it's really, really likely that they are actually related. And there's basically no chance, there's 0% chance that this happened by random chance. The percent identity, it's the highest percent identity for a set of aligned segments to the same subject sequence. What does that mean? So that means that it's what percent of the nucleotides were the same, basically. And those are the 5 things that we're going to look at. You can see that the query cover is really large. The sequences are really identical. And so that means that very few or even no nucleotides are outside of a perfect match. Like these are pretty perfect matches. The sequences are identical or they're extremely similar for the top 5 hits. looking at number 3, what kind of trends do we notice? And so with the exception of a couple of things, they're almost all designated as isolates, which means that they were obtained from human patients with COVID-19 at the healthcare centers. Um, if we were to open this up, um, we might see some non-human ones, like this one here is not an isolate, it's the complete genome. And so it's someone has sequenced the genome of the virus not as an isolate. Um, and we see here, this is a synthetic construct clone. And so this is maybe something that we would use to make like a vaccine. And so it's a lab polyprotein, means that it's made in a lab. And you can see a couple of other synthetic constructs as well. We're creating this genome synthetically, or we were, to um, try and create the vaccine that we created. The most similar non-human viruses. So we're kind of overwhelmed with the data now, now that this has been going on for a year. But back in May, the most similar non-human viruses were from bats and pangolins. And so these are two different animals that have viruses associated with them that are very similar to the human variant that was infecting humans. Um, and though there might be some others that appear, it's widely reported in the media that it may have been transformed from bats to humans, transferred from bats to humans. It's also been recorded that pangolins naturally host the virus as well. And so these are um, the two kind of options for maybe what virus has mutated to be able to infect humans. And so a virus that infected pangolins or infected bats might have randomly had a mutation that allowed the proteins on the surface of the virus and to allow the virus to enter human cells rather than bat cells or pangolin cells. If that bat or pangolin that had the virus comes in contact with a human, it would then transfer to that human if it was able to. And so if this mutation happened and there were no humans around, it wouldn't matter. But if there are humans around, it will get it. The human, I'm going to add, does not need to necessarily eat the bat for this to happen. That it was a really widespread story that is false. And so literally just being around them is enough. I'm going to go back and redo this blast, and I'm going to expand the survey to 5,000 instead of 500 and see if I can identify some of those non-human strains for you. And so we tried 5,000 and it was still all humans. And so the database is just overwhelmed with human data at this point. But if we had done the search back in Maine, we would have, back in May, not in Maine, although I was in Maine in May. If we had done the search back in May, then we would have found some pangolin data and some bat data. And so keep that in mind. Now go ahead and perform section 2. I'm going to do a walkthrough of the different steps of plugging things into the websites. And so if you get confused, come on back and watch that. If not, go through the second part, answer the questions, and then come back and check out what I have to say. So when you go to NextTrain, it'll look like this. What you're going to want to do is go ahead and click on this first link, the latest global SARS-CoV-2 analysis. You'll see that this is updated daily, and so this is really recent information. Click on that, and it's going to take you to the phylogenetic tree. Don't be scared by what pops up. It's a lot of information, and we're going to play with the settings a little bit to make it make more sense, because this is just a ton of data, right? So we're going to do clock, as it says in our instructions. Interesting. We're going to do clock and then branch length to divergence. And then if we can find map options, we go to country. And then under the transmissions map of the world, we're going to select play. So here is our transition map of the world. Let's see here. Click on it. Move this around a little bit, see all the different things. There are lots of different colors. Let's select loop animation to see how this goes. I'm just trying to, and here we can see play. And so we're going to play from December of 2019 to, I'm recording this video in February, so February 20th, 2021. And you'll be watching this, so I'm going to pause and resume after the thing is done. You might also find it interesting to change the color by function to country instead of by clad. And so you can see what's happening in each country, and eventually it'll show you transmission between countries. In order to make that happen, you're going to want to come down over here, and under geographic resolution in the map options, you want to show transmission lines. And you should be able to see that eventually. I'm going to reset here and see if I can get it to play. And so this is an example that was given with the case study. I couldn't figure out how to get the transmission lines, but you can see how the branching, you can see how it's transmitted around the world. And so this is a really interesting piece of data. The purplish colors are the strands that originated or were found in Asia. And then we have blue in South Asia, red in the US, and green/yellow in kind of Europe. And so this is really interesting data to take a look at and see how things were spread in the world. So keep this in mind as we go through and answer the questions. And so if you haven't already, take the time to go through and answer the questions now, and then come back. Okay, so let's think about what these dots represent. Thinking about our blast that we just did, the individual dots are those individual isolates. And so they are kind of those cases that were isolated, sequenced, and put into the database. The lines show relatedness. So thicker lines are going to mean they're more related. Changes happening over time, and branching showing when one strain mutated and led to two or more different strains. And so you can see how they're related, how they traveled, and how the different strains arose. What do you notice about the mutation frequency? How many mutations have occurred between isolates? General mutation frequency is not super high in the example that that the case study gave. Usually a strain will give rise to 1 to 2 or 3 branches. Then 4, are there similarities and differences in viral strains in a country or between countries? How is this related to the epidemiology of the virus? And so the colors for countries and regions are spread among the various branches of the phylogenetic tree. And what that means is that the infection within a country usually has a mix of sources. The European isolates in particular have spread out pretty widely, and have traveled to many other geographical locations. And so this is likely related to travel and how travelers brought different cases into the countries that they were traveling to. Go ahead and try part 3. If you need to walk through the technological sides of things, stick around and I'll show you how to do that. If you want to take it on your own and check back, then go ahead, do that, answer the questions, and check back. So now we're back with BLAST, and we're going to do another nucleotide blast. And so I've copied and pasted the sequence from our patient into here. And so we are going to see what kind of matches we get. I believe in this case, we are going to search specifically beta coronaviruses. And so we want to navigate here and we're going to search beta coronavirus database, which should be at the top. One second. And when we navigate to Nucleotide Blast, we should be able to choose the beta coronavirus database. And so we're going to click here to beta coronavirus and the beta coronavirus gene bank, and we're going to blast this. Let me just make sure we don't change any other settings. Nope, all the default settings. And so we are going to push blast. I'm going to pause while this goes. So we have some results for patient 1. If we look here, oops, it's still working. So we have some data here, and we can see that all of the results come from the US. And so we can think about what that means in terms of the questions. I'll answer the questions now. And so if you haven't done that, go back and do that for yourself, and then we'll talk about it. And so this first patient probably obtained the virus in the United States. And so they probably got it in the US and then stayed in the US as they traveled to DC. As we're looking at patient number 2, the results are a little bit less clear. And so we see a few US, we see this isolate of the genome. And then we also see, if we scroll down, a couple non-US. Really gotta scroll down, but down here we have a couple from Pakistan. We also, you'll notice that the percent identity is only 99.8 or 99.9. So probably a mutation has occurred since these matches, which is interesting. And so this is kind of just demonstrating that the data only gets you so far, right? And so we can compare sequences all we want, but really it's hard to tell where the virus came from or where were the super spreaders, what was happening, where did it start, who did it start with. And so having all this data actually does not allow us to identify a patient 0. And having the complete genome sequence, which we have now, actually biases these results towards patient 0, and it doesn't give helpful information because whatever we've decided is the genome is going to be more related to one strain or another. And so it's not super helpful in trying to find patient 0. Patient 0 is also a historically problematic notation. It started, patient 0, the first patient 0 was in reference to the HIV and AIDS outbreak. And it caused a lot of discrimination against gay men, and in particular against this one gay man who was denoted on a chart as patient O, meaning that he was from outside the United States. Someone interpreted that as patient 0 and that he spread it to everyone else. And so there were lots of discrimination based on kind of assumed promiscuity and all sorts of things. And it was really, really harmful to the queer community and especially gay men at the time. Um, so patient zero, not helpful, all in all. Um, Number two, given that both of these patients presented in Washington, D.C. after flying in, um, what might you suggest? Some things that you might suggest are prohibit individuals from flying internationally. That's something that happened. Quarantining travelers, that's something that happened. Enforcing hygiene standards, we know all about this. Hand washing, wearing a mask, social distancing is something to think about. And also enforcing that individuals need to stay home when they feel sick. For number three, based on your recommendations, reflecting on the outcomes, there might be increased social isolation and anxiety and a toll on mental health in general. And I think that a lot of us have been feeling this over the past year. I know that in the winter, when we were completely online, it felt really isolating to be teaching and not actually seeing you all every day. And so it was hard. And I have lots of friends who are moving out of cities where they feel really isolated to move back home with their families and to at least be close with some people. Last spring, when this all started, I went and spent time with my parents because being alone and or even with just one other person can be really hard. Um, and so those are the social impacts. We've also seen really drastic effects on the economy and thinking about unemployment levels and everyone who's lost their jobs and the different economic blowbacks of the virus. Fortunately, we've seen a lot of improvement in those realms, but we're still not where we were before the pandemic. And so it's going to take a long time for our country and the world to recover from these things. Um, Thank you for going on this little adventure with me. I hope you enjoyed some of the bioinformatics tools. It is fascinating how much you can do with these online resources, and they are just available to everyone. You can go and search whatever you want. If you want to know how related a human protein is to a banana protein, you can go for it. And so if you are interested in this, feel free to reach out to me. I am a huge bioinformatics nerd. I love working with this kind of thing. And feel free to play around on your own on the websites because that you do have access to them and you can compare whatever you want. And so have fun with it. I hope this was a little bit fun and that you found it interesting. And we will see you in class tomorrow.